So here we go. All right, so this is for the Unit 3 test review for collecting data. That test will be on Thursday. And this first video is just going to be going over some basic definitions that we need to know and some general concepts. It's really important that you understand the concept of a good sample. A good sample is one that represents the population. It's also something you need to know is that samples produce statistics, that's the measure of the sample, whereas populations produce parameters, the measure of the population as a parameter. One of the big things is sampling error, and this is something that you're going to see for the rest of the year on. And it's error that occurs because we are using a sample statistic to estimate the population parameter. This error is not bias. It's not. This is not bias. This occurs naturally. Whether a sample is small or large, it will always be there. However, sampling error or sampling variation can be reduced by increasing the sample size. Bias, on the other hand, is overrepresenting or underrepresenting part of the population in a systematic method. Bias means that the design or the study is flawed to favor one or more groups over another. Not necessarily intentionally, but the design is flawed in such a way that some group is going to be over or underrepresented. Bias data cannot be fixed, so increasing the sample size will not help you out. So you need to throw away bias data and start over. So types of bias, or most common types of bias that we talk about. We talk about undercoverage bias, and that occurs when there's a part of the population we're not able to reach due to the design or the execution of our study. We mentioned that in the 2020 census, they start off by sending emails and letters to homes. Well, the homeless don't have that available to them. So obviously there's going to be an undercoverage bias. The homeless will be undercovered. So there'll be undercoverage bias for those who are without a home. Non-response bias, I think, is probably one of the most difficult things to overcome in our society. And it's because we've become cynical and we don't want to respond, or we're too busy to respond. Non-response bias assumes that I actually have a good design for my study, but my execution prevents me from collecting the data. So I have a random selection process in place. My random selection ostensibly represents the population. However, the population chooses not to respond. One of the things that I think of at Reagan is the PTA packets. They go home, or let me rephrase that, they go in the backpack, and there they stay for an entire year. They don't come back. Used to be that when people would call, people would actually answer the phone and answer surveys. Nowadays, that's not so much. So non-response bias is a major issue today, and basically it is a portion of the population choosing not to respond, even though they were randomly selected. Voluntary response bias involves no randomness. That is the biggest issue with voluntary response bias. There's no random selection. There's no people, the people were not selected to participate. They self-selected. They chose to participate. This happens with call-in polls you see on the internet. When they sit there and they say, hey, which song is your favorite? And it's just everyone who wants to call in can call in. And what happens is, if you're calling in for a song, it's either because you love it because it brings a good memory, or you hate it because it brings a bad memory. All right? So basically, voluntary response bias, not only were the people not randomly selected, they chose to participate as opposed to being selected. It overrepresents those with extreme opinions. It overrepresents those who are in favor of something and those who are against something. Those in the middle who are meh, they don't participate because they're not motivated to. They're not motivated to fill out that poll. They're not motivated to call in and talk about that song or that TV show. etc. Response bias results from questions that are embarrassing, sensitive, or non-neutrally or poorly worded due to the phrasing, and they tend to lead people to a particular response. We minimize response bias by carefully field testing the questions. That said, on sensitive topics, there's a lot of times that people will not give an honest response. And the one that I come to that probably hits close to home is when you wanted to go out and your parents had asked you if you'd done your chores or if you had done your homework. And of course, the answer was always yes, even though sometimes it meant, well, not quite, because you wanted to go do something. All right. One other thing, and by the way, there's a massive typo on this particular line, but when you're answering FRQs, it really helps if you begin with a simplified definition that shows that you understand what the vocabulary word they're talking about. And then you must explain it in context of the problem. If you just give the definition, you're not getting credit. However, if you give the definition and you give it in context, you're likely to be given the benefit of the doubt if the context is close but not perfect. But by giving the definition, you tell the reader, which in this case is Ms. Lamb and myself, that, hey, I actually know what this is. My English may have been a little bit sloppy because I was in a rush of the test, but it's close. So you may be looking at getting a partial credit or perhaps even a full credit, depending on what you have. Observational studies is one of the ways we collect data, and they are observing people doing what they already do. In doing so, with an observational study, they are basically self-selecting. We are not assigning them treatments. And they are really good. They're the most common studies we have. I mean, and they can show correlation, they can show association. They may show that there's a relationship, and they can be statistically significant, but they cannot show causation. Most studies are observational studies. Please remember, historical records and medical records are also considered to be observational studies. Once again, observational studies, the treatments are not assigned. Surveys are a type of observational study, and you need to be able to recognize and conduct the following types. Make sure whenever you run an experiment or an observational study that you always address assignment, always to say how they went to a group, all right? So in a census, basically we're counting everyone in the population. If we do that, there's no need for statistical inference or techniques because the population parameter is known. So if we conduct a census, the entire population parameter is known. Most of the time, a census is not something we can do because it's too large or there's other difficulty in getting the data. But there are times that we can. If you want to know how many math teachers at Reagan, I don't need to take a sample of teachers to find out. I can go count them. I can find my class average. So there's a lot of times that I can do a census, but when we have large populations, it becomes difficult. And my favorite example of when it's virtually impossible is when I want to know how many fish are in a lake. Well, the only way I can do that to really conduct a census is the drain of the lake, in which case there's no lake and therefore there's no fish. So it doesn't really work. The simple random sample. This is the basic of all samples. And whenever I see it, I think of one thing: put everyone's name in a hat and mix. That is not the only way to conduct it. You can assign numbers, because like if I had a shelf full of jars and I wanted to randomly select them, I would probably number the jars, 1 to whatever, put those numbers in a hat, and then select the jars that were chosen. Please remember, there is no special case or subset of simple random samples. Stratified sample, maybe Mr. Spencer's favorite, but it's not a type of simple random sample. There are no types. It's only the simple random sample. And this is really, really important, and it's not written here. Actually, it is. Every individual has an equal chance of being selected. That's true of all good random samples. The next part is not. Every group has an equal chance of being selected. Every group has an equal chance of being selected when we're talking about a simple random sample. And that distinguishes it from all other types. A systematic sample. The systematic sample also involves randomness, and it involves randomness before we pick the first person. So in a systematic random sample, every individual has an opportunity being selected. However, not every group is possible, because once you've selected the first one, then it's going to be every nth term after that. It could be every 50th or every 100th or 1000th, depending on how rapidly we needed to make selections. The cluster sample and the stratified sample. Those two are the ones that cause the most trouble for my students typically, or for the most trouble for students. So I want to spend a little time on them. With a cluster sample, we are going to break the population into smaller groups. And those little groups are microcosms of the population. In other words, a good cluster should look like the population. It should have all the elements of the population. That's why when I draw blood, that's essentially a cluster, because that little sample of blood represents all the blood in your body and has the red blood cells, the white blood cells, the platelets, the plasma, and whatever is healthy and whatever is sick should be represented in the sample. So cluster samples, we have little microcosms that look like the population. Then what we do is we randomly select one or more of those groups, one or more of those microcosms. Once we've selected them, we then survey every single one in it. Now, hear me out. Sometimes my students go, you know what? I couldn't tell between a cluster or a stratified. How can I help? What can help me here? So this might help you just a little bit. If you've narrowed it down to, it's like, hey, it's groups. We have some sort of group. We have a cluster group and we have a stratified group, but you're not sure which one it is. I mean, obviously, if you know it's cluster and stratified, you'll be fine. But if you didn't know what it is, this might help you out. So hear me out. If I have groups and then I have to survey every single element in the group, it's a cluster. So if I have to take a group and then perform a census within the group, that is a cluster sample. All right? The next one, the stratified sample, which happens to be my favorite. To perform a stratified sample, the population is once again divided into groups. But they're divided into groups based on a common characteristic. The groups are called strata. So the easiest one for us to visualize as high schoolers is freshmen, sophomores, juniors, and seniors. So that would be those would be my strata. There would only be freshmen in the freshman group. There'd be only sophomores in the sophomore group. There'd only be juniors in the juniors and only seniors in the seniors. And when I run a stratified sample, I select I'm going to go to every single one of the groups. There's no randomness in going to the groups. But within the group, I randomly select. All right, so I'm going to randomly select within each group. And the nice thing about this, one of the nice things is it minimizes variation in the proportions of representations. So one thing happens when we do a completely randomized design, a completely randomized sample. It is possible, or a simple random sample, it's possible that I put all the students' names in the hat at Reagan. And when I did that, it's possible that I drew no seniors. That'd be very sad for the seniors. However, if I do a stratified, that will never happen because I will draw from every single group. Okay, so we minimize the variation proportions of the populations because we are taking from every single group. Experimental design. Treatments must be assigned. You must say how they are assigned. So that means I put the names in a hat. I blocked whatever you needed to do. You must say how you did that. And that's what sets experiments apart from observational studies, is treatments are assigned. Remember, experiments may show causation, but they are costly, time-consuming, and often limited by ethical constraints, which is why observational studies are more common. But experiments can show causation because treatments were assigned. Treatments are not assigned in observational studies, and they cannot show causation. Very important that you know the purpose of blocking and be able to determine on which variable one should block. We'll do some examples here in a little bit. You need to be able to design all three types of experiments: the completely randomized, the randomized block, the matched pairs. That's not to say that you will necessarily have to do each one of those. It's going to depend on the test you take. Confounding and lurking, those two words are essentially interchangeable. Do not worry about a difference between them. All right, so I do not want you to stress about there being a difference between them. What I do want to say about when you have a confounded, it means you don't know what is causing the change you're seeing. So something that's confounded is because we don't know what's causing the change. And the lurking is the underlying variable doing it. So they're essentially, if you swap them out on the test, you're not going to have any issue with us. How do we combat confounding variables? This is why you do random assignment. When you do random assignment, we're less likely to have it minimizes outside variables. So random assignment minimizes the effect of outside variables. So random assignment minimizes the potential of having a confounding variable. One of the questions was, will we have to explain confounding on the test? And that's a good question. You might have to come up with a variable that you think might be a confounding variable. As far as defining confounding, no, you wouldn't have to do that. But you may very well have to identify something that might be confounding, and you'll get an opportunity this evening to see that. You need to know the purpose of a control group. You need to know that the purpose is to provide a baseline so we know whether or not the changes we're observing are due to an outside source, an outside variable that wasn't controlled for, or due to the treatments that we imposed. Know the components of an experiment. Know that this is going to be huge. Know that generalized, I can't even say the word, so I'm going to let y'all read it, is the purpose of the sample, and that can only be generalized to a similar population. So know that when you want to generalize, if your sample only has males in it, I can only generalize to males. If your sample only has females in it, I can only generalize to females. If the females are aged 25 to 35 and live in Texas, I should only generalize to females aged 25 to 35 who live in Texas. Please make sure that your sample is generalized. Only the population is generalized to matches the sample. All right? And this right here, every single year gets some people. That first word is causal. The first word is causal, not casual. Every single year I have people fall for that on a test. They read it as a casual relationship as opposed to a causal relationship. And I have to say that, being the cruel individual that I am, that's the one time that I make a fair amount of fun of people for it, because I tell them it's coming. So please know that causal, and a causal means that causal relationship can only occur if we're conducting an experiment. All right, so I'm going to stop the recording.